So let's talk about genetic algorithms. I'm in a kind of ambiguous emotional relationship to genetic algorithms. I love molecular evolution. This is my current passion, so I read everything I can about it, and I think a lot about it. On the other hand, as a machine learning technique, I'm less attracted to it than to other techniques, and I'm a little more skeptical of it. So I'm not sure how to reconcile the two. I mean, genetic algorithms, as were used by nature for several billion years, have done amazing things. So I guess there's every reason to think they could do amazing things for us in the lab. Anyway, so let's talk a little bit about what genetic algorithms are in nature. So I assume most of you know most of this from general education, but I'll just put it on the board and talk a little bit about it. We all have genomes, which are long molecules. For simplicity, we'll assume it's one long molecule. It's really broken into chromosomes. In humans, 23 of them. In other animals, a variety of numbers. But they can be all put together conceptually into one long molecule that contains a lot of information. This is DNA. And the molecule is really used as a repository of data. The data is, you can think of it as code, different kinds of code. And it's written in an alphabet of 4 letters. A, C, T, and G. You can think about them as 4 different chemicals. And the actual chemical properties are not that important. It's pretty clear that for the most part these are used just as a template to derive other things. And since there are 4 of them, very roughly speaking, every position gives you about 2 bits of information, if you want to think about it from an information theory point of view. Okay, so you have 2 bits of information per position. Those of you who remember our information theory lecture should point out that it's only 2 bits if they're uniformly distributed, and it's less if they're not uniformly distributed. And they're not uniformly distributed, but it's close enough. So how many bits of information are in genomes of different organisms on Earth? Let's start with the smaller ones. Viruses have in the order of 10 to the 4 bits. An HIV virus has about 10,000 positions, so about 20,000 bits. Influenza viruses are similar in size. Some viruses are even smaller than that, like the smallest one is 4,000, 3,000. And if you think about the entire program of the virus written in this code, it's quite amazing. Now, I didn't tell you what the program is, so let me tell you a little bit. For simple organisms like viruses and bacteria, the code is pretty dense in the sense that every segment of it encodes for something pretty. It's very clear to us what it encodes for. So typically, there would be positions here that encode for proteins. And these in between regions are control structures that tell you when to encode this protein, under what conditions, and how much of it to create, and so forth. But it's basically a recipe for making proteins, and proteins are complicated molecules that act as machinery in the cell and has lots of other things. So viruses have on the order of 10 to the 4 bits, and on the order of 10 proteins. And again, it varies. The simplest virus I'm familiar with has only 4 proteins. Complicated ones can have dozens or maybe over 100. But this is the rough order of magnitude. Compare that to bacteria. I should say that proteins are pretty much the same as genes. Each one of these is a gene. So gene is a recipe for a protein. Bacteria have on the order of 10 to the 3, 10 to the 4 genes, proteins, and on the order of 10 to the 6 or 7 bits in their program. So they're significantly more complicated than viruses. And then people like to think of us as like the epitome of creation, but actually there are plants that have more genes and more bits than we do. Okay? There are lots of plants that have, and there are bugs that have more. So when I say we, I mean all animals and all plants. But I'll give you numbers based on people. Current estimate for people is about 30,000 genes. And we know exactly how many bits are in the program. There are 6 billion bits. Or 3 billion of these positions in the DNA, in a human genome. I write here genes, but actually the number of protein that makes could be significantly larger. It could be 100,000 proteins, or even more, because when you deal with animals and plants, as opposed to bacteria, the mechanism of turning the gene into protein is much more complicated. The genes actually are distributed programs, and there are different ways of combining them and mixing and matching and excising parts, and so one gene can give rise to many, many different proteins. Some genes give rise to one protein, and other genes give rise to thousands of proteins. They're somewhat related. So this is just to impress upon you the size of the programs. And I imagine that these programs, they're very sophisticated, and they were created by some kind of a random process, by a blind process, which is really what evolution says. These programs came about by a random process of trial and error. Now think of this space, the state space, the space of all possible human genomes. It's this long line, if it's of length 3 billion bits. I'm sorry, 6 billion bits. If it's 6 billion bits, the number of possible such strings is 2 to the power of seems right. Right? This is how many strings there are that are bit strings that are 6 billion long. And if you think of each one of us representing a point in that space, and us as a species overall being concentrated in a very particular area, right? If you take a random bit string of 6 billion bits, the chances that it encodes for a functioning human are null, right? But how in the world did nature find that particular place where we can function? Compare that to my favorite number, which is the number of elementary particles in the universe, which is about 10 to the 80, which is the same as about 2 to the 250. These are huge numbers we're dealing with. So we all think that we know how evolution works, and the idea is that it's sort of a trial and error search, that you start somewhere and then there are mutations and random variations, and then those mutations that lead to higher fitness result in people who carry, or animals who carry these mutations, reproducing more, and in the next generation there are more of them, and as a result mutations that are advantageous, that increase our fitness, will tend to spread in the population and take over, and therefore the population as a whole will be moving in this space of strings towards strings with higher and higher fitness. That's the sort of high school version of evolution. The reality is a lot more complicated, and it has to do with the fact that the samples are a finite size, and that the steps are stochastic. So if you take an extreme case where the population of an evolving species is quite small, let's say it has only 1,000 individuals, and one of them has a mutation that increased its fitness by 5% relative to the others, which is quite a big improvement. Maybe they got stronger muscles or something. Still, chances are that mutation will disappear, much more likely than not. On the other hand, somebody else got a mutation that didn't do anything to the fitness, just changed their eye color or something that didn't really affect fitness at all. There's still a chance that it will actually propagate in the population, a non-negligible chance that it will propagate in the population and eventually, after enough generations, fixate in the population, which means will take over everybody in the population will have that mutation. So there's a significant level of randomness, stochasticity in the spread of mutations in the population. That significant level goes down as the population size grows and grows, but it turns out because of patterns of reproduction that what matters is not the overall population size, but what's called the effective population size, which is in reality much smaller than the actual population size. So it's actually quite a complicated process of figuring out and understanding how mutations evolve and propagate, and we're very far from having a clear understanding of why things happen the way they happen, and I think the whole field of natural evolution is still ready for another Darwin or two. There are lots of things that are not clear, which is not to say that I support the alternative. Absolutely not. I don't think the alternative as presented is scientific, but as far as scientific theories go, this is a wonderful theory, just like Newtonian mechanics is a wonderful theory, but it still needed some tweaking. And I believe that we still need some tweaking and understanding of how evolution works. Anyway, let's go back to how it's related to machine learning. So let's abstract away the mechanism that we believe is at the heart of natural selection and evolution, and see if we can sort of benefit from it for learning in a similar way. So here's an algorithm for looking for a hypothesis, trying to find a hypothesis using a genetically inspired mechanism. I should say explicitly, when I talked about this huge space of strings and how evolution searches it, really what we're talking about learning here is a search problem. Namely, given any DNA string or given any hypothesis in our simplified world, we assume that it is a straightforward matter to figure out its fitness, how good it is. The problem is finding hypotheses that are as good as possible. Okay, so this is the part of machine learning that is strictly a matter of search. It's a huge space, it's an exponentially large space. Our problem is not assessing individual candidates, we assume that this is trivial. Our problem is finding some candidates that are as good as possible. As you think about it as a search problem, I'd like you to think, what are the characteristics from a computational point of view, what are the characteristics of the kind of search that genetic algorithm does? I mean, you could do another search, right? You could just systematically search through all the strings. I mean, you'll never finish, but you could choose some subset of the string and search through them. You can do some local search, you can think of gradient descent, you can think of all sorts of searches. Think about what we do, the algorithm I'm going to show you as a search algorithm, and I'd like you to tell me what are the characteristics, what are the properties of that search, good and bad. So here's the search algorithm, call it GA, it's a genetic algorithm. We're assuming that there's a large population of possible hypotheses, H, and that for any one given hypothesis, we can immediately evaluate its fitness. And our goal is to come up somehow with one or more hypotheses that have as high a fitness as possible. It's a good question to know when to stop. Do we know what the maximal fitness value is? Sometimes we do, sometimes we don't. So sometimes you don't know if you got as high as you could. It's a little bit like in neural networks when you're trying to reduce the error rate and you don't know if you've done the best you could. But anyway, the algorithm takes as input a fitness function, a fitness threshold, which is going to be used for deciding when to stop, and some numerical parameters, P, R, and M, that have to do with the 3 steps that are going to be used, actually more steps, in the genetic algorithm. So we'll talk about them as we go through them. We start by collecting some random subset of the hypotheses. Remember, there is an exponentially large number of them. We're going to collect a finite sample, maybe 1,000, maybe 10,000, as many as we can afford computationally. Put them into the P variable. Then for each hypothesis in P, we calculate its fitness. We said that's trivial, right? For any given candidate, we can calculate their fitness. So we have the fitness of all of them. And then we enter a loop, and the first thing we ask is have we found what we want yet? Namely, is the maximum fitness in our population, P stands for population, is the maximum fitness of any element in our population good enough? And assuming it's not good enough, so we are here now, we're going to select parents for the next generation. So we're going to try to select some subset of our current population to serve as parents, as progenitors of the next population, and we want to select parents that are as fit as possible. So we're going to select parents proportionately to their relative fitness. So we calculate the relative fitness, which is the fitness of each hypothesis divided by the total fitness, and we're going to use that to select a certain fraction of the population that would be used as parents. The fraction is 1 minus r times p. Then when we selected the parents, we're going to replicate what happens in most sexually reproducing organisms, which is we're going to do a crossover between the genes or the programs representing one parent with those representing the other parent, and the offsprings are going to be a mixture of the two. In real biology, only one particular mixture is chosen. You know, when your father and your mother's chromosomes mix and crossover, one combination was chosen, the other one is discarded. In our game here, both children will be chosen. So when you do a crossover, you get first part from mom, second part from dad, one child, and then first part from dad, second part from mom, the other child. Our crossover can occur more than once. So we select, again probabilistically, this many pairs of hypotheses from the population, and for each pair we produce not one but two offsprings by applying the crossover operators. And then we add the offsprings to the mix, and then we apply mutation operations. So all this is inspired by the way things work in real biology. First there's crossover in generating the next generation, and then there are mutations. Just to give you some numbers, mutations in creatures like us is about 10 to the minus 9. That means every particular position in the DNA has a chance of 1 in 10 to the 9 of being changed. And if you remember, we have 3, I'd say 10 to the 8, 10 to the minus 8 and the minus 9. And since we have 3 billion positions, that means that every time our genome is duplicated, we introduce on the order of 10 mistakes or 10 mutations. Okay, so that's not very much, and most of them don't do much. But over time, over many people, over many replications, some of them do interesting things. Most DNA-based life forms have this kind of replication error rate, which is quite remarkable for a messy chemical system. And it's because of a very, very sophisticated mechanism of repairing So actually mistakes are made much more frequently than this, but there's a mechanism that detects them and repairs them. Some life forms, RNA-based life forms, basically viruses, specific kinds of viruses that are based on RNA, have a much higher mutation rate. So HIV, virus responsible for AIDS, and influenza are both RNA-based life forms, and because of the chemical differences in how RNA is replicated, because of the way they replicate RNA is much more error-prone, they make mistakes about 10 to the minus 4. Which is a huge difference. Now remember that viruses have smaller programs, and their programs are about 10 to the 4. So they again make about 1 mistake or 5 mistakes per replication. But they replicate much, much faster than we do. So as a result, these viruses go through evolution and natural selection at a dramatically faster rate than we do. We, other animals, plants, bugs, go through natural selection at a rate of, over millions of years. You see changes that take literally tens or hundreds of millions of years to see in the animal kingdom. You see with these viruses over a period of weeks, months, years. The differences are dramatic. So in a way, the entire creation, in a way, is replicated by these viruses. The story is told again and again by these viruses every several weeks. In fact, a single one of these viruses infecting a single patient grows very fast, within a matter of weeks, into billions of individual variants, each different from every other. Because every time there's replication, there are a few mistakes made. The rate at which they proliferate is phenomenal. One of them infects a cell and then can make anywhere between 100 to 100,000 other particles coming out. And most of them are not good particles. They're not able to replicate because the mistakes were the wrong kind. But maybe 1% of them survive. And as a result, within a few weeks to a few months, you have a whole species of viruses living in your body. Hopefully not in your body. So there's really evolution and natural selection happening in each individual person as a whole universe. Anyway, don't get me going. I told you this is my passion. Okay. We are at mutations. So we invert a randomly selected bit in this proportion of the members. So M is a number between 0 and 1 that stands for mutation, and PR and M, what is R for? Reproduction. Thank you. Okay, and then the next generation becomes the current generation, and then we evaluate the fitness of every member of this new generation, and we go back to ask, have we settled yet? Okay? So this is a simplified view of natural evolution, but it's very easy to implement in a computer program. What you do need from the outside is this ability to evaluate any arbitrary member, any arbitrary candidate with regard to fitness. So you need a function. That's the one called fitness. That given a hypothesis tells you its fitness. And then you can go to work on finding hypotheses that have good fitness. And then once you've found one that has higher than fitness threshold, then you return that as your answer. So what do you think about this algorithm as a search algorithm? Well, you can tell me if you think it's good or bad, that too, but what I mean is, what can you say about it? What kind of properties does it have, thinking as a computer scientist? Please.
It works well if combinations of two fit strings also are a fit string. If you've got some complex space where you've got two local minima and you mix them and it's bad, then the search algorithm isn't going to work great.
Yeah, that's a very, very good observation. It assumes, or it will work well, if there's some kind of a compositionality or additivity of features for fitness. So it will work if generally, not always, it doesn't always have to be the case, but it should generally be the case that if there's some aspect of this hypothesis that's good, and there's some aspect of that hypothesis that's good, then together they have some chance of being even better. Which is generally the case in most reasonable representations. Of course, the counterexample that immediately comes to mind is, what's the counterexample of all machine learning? Parity, XOR, where nothing has a clear positive or negative meaning separate from the context of everything else. Right? So parity will not be learned well by this mechanism, because it's like the quintessential, you cannot combine things and they retain their positive value. So to turn it into a biological interpretation, if strong muscles are good and big brain is good, then strong muscles and a big brain is probably even better. Okay, but there will be exceptions, that's okay. But generally this should be the rule, that a good feature and another good feature together are even better. This is an assumption made here. So that's a good point. I was looking for something even simpler than that. Is this algorithm guaranteed to converge? How long will it take? Is it parallelizable? Is it deterministic? Is it any of these things? So
it's sort of like a parallel local search with some jumps.
That's a very good characterization. It's a parallel local search with some jumps. So what do we mean by that? First of all, it's a parallel search. That is an important thing to note. At any one point, the population represents a set of candidate solutions. And that set could be distributed over this entire space, the space of all possible solutions. And each one represents a possible search area. And then the crossover operation does, I think what you were referring to as the jumping. So if you can think about it visually, one population corresponds to these points. And you could stop at this population and then do local search around each one of them, right? You can take each one of those hypotheses and make small changes like by mutations and do some kind of a gradient descent or a local search around them. But that's not what we do. What we do is we pair them up and jump to some points in the middle. That's what the crossover operation does. So there are these jumps. But it's still the case that this is a parallel search in the sense that it entertains many possible solutions at any one time. Contrast that with something like traditional gradient descent. At any one time you're at one particular point and you move gradually, incrementally from that point, maybe follow down a gradient or something. Or contrast that with EM. How does EM behave in that regard? Is it one point or many points? Does EM algorithm keep track of one possible solution at any one time or many possible solutions? One. Does it make incremental changes or jumps? Jumps. Okay, so EM is somewhere in between. EM does keep track of only one, but it can jump. The M step, unlike gradient descent, which makes small changes, the M step can jump pretty far. So this has characteristics of EM in the sense that it jumps, but it also has the characteristics that it does it in parallel with many possible positions. Other differences is along the determinism. In EM, once you chose your starting position, it's pretty much predetermined where you're going to end up. It's a deterministic algorithm. So is forward-backward. So is backpropagation. They're all non-deterministic in the sense that you can start anywhere you want. Well, it's not. I wouldn't call it non-determinism. They're deterministic, but they do depend on where you start. Genetic algorithm is not. It may be the first algorithm we are discussing, which is randomized. Is it true? We discuss other randomized algorithms. You can randomize many algorithms, but we didn't do it for decision trees. We didn't do it for neural networks. So I think it's the first one we're discussing that's randomized. So it's parallel. It's a parallel search. It's randomized. Not just starting conditions, but every step you take has some probabilities, so you branch in different directions. So starting from the same position, you end up in different places in different days. Randomized jumps as opposed to taking small steps. And it makes the assumption, or the underlying assumption for it to produce good results is compositionality. What I call compositionality, which I explained earlier, is this notion that one good thing and another good thing, you put them together, most of the time they should give you something even better. Okay. We didn't specify everything you need in order to implement something like this. The most important thing that's missing is how do you represent your hypothesis so that you can do the crossover and mutation operation on them. So really the remaining part is take your hypothesis space and represent it as a bit string. And what you choose as your representation will affect greatly whether your algorithm does any good. So the choice of representation is intrinsically tied to the dynamics of what will happen, what possibilities would be considered. So let's take something very simple. I would call it even lame. Let's take our famous enjoy sport kind of representation and give you an example of how you can represent it as a bit string. Keep in mind, this is an example. You can think of other ways of representing bit strings, representing by bit string. So suppose we want to represent the condition that the outlook is either overcast or rain and the wind is strong. Well, you can represent each attribute by a series of bits, a bit string, short bit string, one for each possible value of that attribute, and assign one when that value is allowed by the condition and zero otherwise. So this would be outlook is overcast, rain, but not the third one, whatever the third one is. Clear, sunny, sunny. So sunny is not allowed, but overcast and rain are allowed. And wind is strong. Wind has only strong or weak, so strong is allowed, weak is not allowed. And then you can use that to represent if-then statements, which you can think of if-then as a hypothesis. So this was the play tennis, not the enjoy sport problem. And how would you represent if the wind is strong, then play tennis is yes, which is a particular hypothesis, a particular concept. Well, you can represent it by, I don't care what the outlook is, so all values are allowed, and wind has to be strong, and then play tennis would be yes. So the first bit would be for yes, play tennis, the second bit would be for no. Now this is somewhat redundant. You could represent the output with just a single bit. Okay, so there may be a reason why you want to do it one way or another. If you allow arbitrary crossovers and arbitrary mutations, then you have to be prepared to deal with any values of these bit strings. So you have to decide on their interpretation. So what does it mean for it to be both 1-1? Well, does it mean that you don't know whether you play tennis, what the value of play tennis is? Maybe. So there's no magic here. It's up to you. It's a matter of your convention. How do you represent your hypothesis space please.
Isn't that more wind equals strong and play tennis equals yes? Than if wind equals strong, then play tennis equals yes?
Well, that was the case here. It's up to us what it means, and what this is going to be. No.
Two different representations.
Yeah, this is representation of conditions, this is representation of rules. And these bits mean something different than these bits. I mean, it's like put a line here and say, okay, we're going to represent things as on this side will be the conditionals, on this side will be the outcome. The only thing that's important is that you understand your representation, so that when you do your operations, crossover, mutation, or any other operation you decide on, you understand what it does. And it's up to you to stop and think in the abstract space of hypothesis and say, are these the kind of operations that I want to try? Remember, there's no magic here. You're basically doing a parallel search, and you're doing jumps in the space. You're trying different things. And maybe the most important thing is that you're taking advantage of your assumed compositionality. You're saying, okay, I'm going to try different combinations of different things. If I have, let's say, 2 members of my current population that seem to have good fitness, I wonder what aspects of them are contributing to that fitness. There are they have many attributes, and I'm going to just randomly choose some subset of attributes from one, some subset of attributes from another, put them together, and maybe I'll get something better. Well, chances are, if they have many attributes, for you to hit on just the right combination would take many random attempts. So one thing you might want to consider is having many children. Take the most fit male and most fit female, or in this case they're not gendered, so just the 2 most fit individuals, and create many crossovers of many types. That might expedite your search. So we're not restricted by Mother Nature, right? Mother Nature says you can only have so many kids, but here you can do whatever you want. How are we going to do, once we define these bit strings, how are we going to do crossovers? Well, here are examples of, these are the parents, these are the children. Crossover mask is basically just telling you where to cross over. So you see this child here consists of this part from this parent and switching over this part from that parent. And as I said, we don't throw away the second child. Nature does, but we don't. In this case, you get the opposite part. This part from here to here, and then from here to here, you get here. This is called a single point crossover. You can have a 2-point crossover where you go from here to here to here. Here's the mask, and you can have uniform crossover, which means that any bit, this is a random string of bits, and one means that this child gets the gene from that parent, and this child from that parent, and 0 means the other way around. So you just have lots of crossovers, and this does not correspond to anything that actually happens in biology. But you can do that. And then point mutation means that you just choose some position and flip that bit with some probability. So at this point you can forget about biology and just think about it as a search problem, and ask yourself what kind of operators make sense and would be interesting and would be somewhat efficient. And it becomes clear that most of them are everything you come up with is extremely inefficient. I mean, think of the example of 2 fit individuals, and then you're trying to figure out which aspects of them. There's so many possible things that can make one of them fit or make the other one fit. And you'll have to hit on exactly the right combination here, the right combination there, and put them together. So you may be searching for a long time without finding anything. Of course, it all depends on the size of the space. Okay, so even this story has complications. So in real biological populations, it is the case that more fit individuals have more children. In fact, this is how we define fitness. We define fitness by the ability to have more children that survive to the age where they can have their own children and so forth. But even if you think of fitness in terms of killing woolly mammoth or escaping danger or strength or anything else, fitness is associated, has been associated historically with human culture and other animals with having more children. To an extent, it's no longer true today in our culture, but it's certainly true for the first several 100,000 years of our existence as a species. Now, take that to an extreme. Suppose the most fit individuals have a lot of children, and the least fit ones have none. Is that a good thing in terms of our search algorithm?
Not necessarily. There may still be attributes that the least fit ones possess, which the most fit ones do not possess, and could be lost in the search.
Okay, what that would correspond to is we found a few guys that we like, and we kind of forget about the rest. We let them reproduce the entire next generation. So the next generation is all going to be concentrated in this area. Right? That's what it would mean. And your point is that we're going to lose a lot of interesting partial solutions that could have made things better. So this begs the question of how much time do we have to experiment? If we're told that we need to produce our output within the next generation, it might make sense to choose just a few most promising parents and have them reproduce like rabbits, and optimize locally something here. But if we're shooting for the long term, if we have time and we know we're going to be allowed to search for a long time, we might be better off maintaining genetic diversity. Namely, maintaining lots of good ideas and good solutions and good attributes in different combinations here, with the hope that later somehow we will be able to find a right combination that will get even better fitness. So this should remind you of something we just discussed in reinforcement learning. Remember a trade-off? Yeah. Exploration versus exploitation. Nature has its own way of dealing with this dilemma. There are biological factors that limit reproduction abilities of any individual, especially of females, if you talk about mammals or most higher animals, which is probably a good thing from the point of view of sort of long-term survival of the species. It is a good idea for a species to have a large pool of alternative solutions. One of the reasons, which we haven't discussed until now, is that the notion of fitness changes over time in many unpredictable ways. So there are environmental changes, there are changes in the predators or in the food available, and all of a sudden something that was an advantage becomes a disadvantage, something that was a disadvantage becomes an advantage. So in a world where fitness function changes over time, it is even more important to maintain diversity, because that means that the species as a whole can adapt much faster. The filter that the environment poses on the species changes, but as long as the species maintains a large diversity, it has a bigger chance of at least a few of the individuals making it through. Otherwise, your probability of extinction is greater. So there are lots of examples like that. One of the most famous examples from Darwinian evolution stories is the famous butterflies in industrial England, There was a species of butterflies that had, I think, a variety of colors, and with the pollution that industrial evolution introduced to England within a very short period of time, only the butterflies that I think were black or fit into that horrible pollution background survived, because they were able to camouflage themselves. Well, that black color may not have been an advantage before, maybe even a disadvantage. But because there was diversity, when calamity comes, some members of the population all of a sudden are in a position of advantage. You could make the same argument for our modern society with regard to ideas and sort of cultural values, and there is an advantage, an intrinsic advantage in diversity of ideas and diversity of theories. As long as we don't live in a perfect world of mathematical equations, we live in a world with very limited knowledge and limited understanding and constantly changing circumstances. Diversity, for the sake of diversity itself, is very important. It makes us more robust as a society. Tom?
I'm not quite sure that sort of diversity is comparable to the diversity we're talking about in a search space like this.
Okay.
Because that is very, very focused diversity. Like once you have a species, and we call it diverse, like in the entire space of the possibilities of the genome, it's only a very, very small little bubble in that area. And you have points all around that tiny bubble, but you don't have random points all over the place to maintain diversity, because all of those points, they can't even create a human being or anything that would live for 1 second.
Quite true. The diversity is a relative notion. Relative to all life forms, we people are extremely non-diverse. We're all like each other.
Yeah.
In fact, the difference between any two people on the planet in terms of DNA is about 1 in 5,000 positions. So that underscores your point. But there's the other kinds of diversity at that scale, the diversity of species. So if you think about survival of the biosphere of life on Earth, diversity of species is an important thing. If you talk about global environmental changes, disasters, meteors hitting, and so forth. This is a good point, and you may find it surprising that there are far fewer gorillas out there in the world than people. Okay, and yet, if you take two random gorillas out there in the wild, they are further apart genetically than two random people in the world. There's much more genetic diversity among the gorillas than among people. So viewed from the point of view of gorillas, we are all incredibly interbred little branch of the family. We all look exactly the same to them, or almost exactly the same, whereas they are far more different than each other than we are. So diversity is relative. Okay, so I started talking to you about what happens when you don't have enough genetic diversity. This is a real problem with genetic algorithms, and it goes by the name of crowding. Crowding is when a few good fit hypotheses are so good that they tend to dominate the parenting and selection process and reduce genetic diversity, which might lead in the short term to gains, but it reduces your ability to have longer term gains. So you may want to fight it, and you could say nature fought it by a limited number of kids, even very fit individuals can have, but you can fight it in your program by sort of reducing the relationship between fitness and number of progeny. So you can do that by sort of not taking too seriously the fitness function, because fitness function is often an estimate and sometimes an overestimate. You can reduce that fitness function, make it a little bit more uniform. You can do rank selection, which means you rank all the hypotheses by fitness, and then you ignore the actual fitness value. You just assign some kind of function that says the higher the rank, the more likely you are to be chosen, but not by much. So there are lots of things you can do to explicitly control for convergence on the most promising current members versus maintaining diversity to allow you to have, so there's exploration versus exploitation. You can numerically control that in your choice, in your stochastic choice of how to choose the next generation. Then,
but say we choose exploitation indeed, wouldn't, and we kill some features of the lesser individuals, so to say, wouldn't the mutation part still grant that
yes.
that we're going to get the rights in a longer period of time?
Yeah, I mean, as long as you have mutations, you have some chance of getting to any string in the space, but the chances are very, very small, exponentially small. It's effectively, it's the crossovers that make the big changes in the space, and mutations make small changes. So yes, mathematically speaking, you have a non-zero chance of getting from anywhere to anywhere, but practically speaking, you're reducing your chances of real exploration significantly.
We could boost, so to say, the randomized part, I mean
the mutation part. Right, you can think of a genetic algorithm where forget about crossovers and you just have mutations, right?
Crossover, but we generate a lot more individuals by mutations.
Yes, you can. And there are examples from nature where mutation rate is surprisingly high in particular positions called hotspots. And I wonder if that's related. Tom?
It doesn't really seem like anything like crossover exists in nature. Just, I mean, only species reproduce with each other. And
I see.
And the results always pretty similar.
Not true. Not true. Lots of examples of lateral gene transfers. There are genes that have been moved from different organisms. Bacteria often carry genes from other bacteria. Viruses carry genes from one organism into another organism. There are now discovering more and more examples of genes being carried laterally.
Yeah, but that only happens in cases where it doesn't hurt solubility very much. Like if you have an extra gene and it just gets integrated into the genome, or if you have two things which do almost the same function, one can get put into the other other's place. Like they're not large jumps. I don't see any sort of large state-space jumps in nature, really.
Again, it all depends on the scale you look at.
Yeah.
If you look across species, you're right. I mean, there's not much going on between species. However, you should know that current theories, fairly well established, talk about some major events of whole genome duplications. So a more accurate view of how life evolved is that there was a small number of genes and then they duplicated, and once you have two sets of each gene, there's no selection pressure on both members of the set to do the same thing, so they're free to mutate much faster, to take on other values, and then 250 million years later there was another duplication event. So they're actually, people who study now go back in genomes of many different individuals and try to work backwards, trace back when those gene duplication events happen and how they happen. And then there's a further, there are some big events, and they happen actually not so rarely. Chromosome reshuffling. When you have speciation, when one species becomes two, often the two species have different number of chromosomes. They started with the same gene pool but broken in a different way. This one had 15 chromosomes, this one had 17. I'm sorry, they all started with the same species, 15 chromosomes, and they split, and this one now has 17 chromosomes. And then the chromosomes might have, this new chromosome can have half of this chromosome and half of that chromosome. So you see evidence of actual, if you compare human and mice, this is one of the most fascinating computational analysis was done in recent years with the availability of whole genome of people and mice and other animals. Human and mice split about 80 million years ago. It's around the time that most mammals split. If you go back and look at the very high-level view of human genes and mouse genes, there are many, many human genes that have clear analogs in the mouse and vice versa. And it's clear that they came from the same source. And you can try to start piecing together how that split happened, what was the common animal, and which genes split and then duplicated and split again and so forth. And there were quite a few such events along the way. So it's nothing like the events that happened every time you have a child. It's not at that scale. It's more the scale of speciation, which is something that happens every few million years. But it's still a very meaningful. We wouldn't be where we are now. In fact, all the wonderful solutions that we found as humans, our genes found, were not really found by us as humans. They were found way before there were humans. So take our immune system, which is an incredibly complicated, ingenious system. it wasn't invented by natural evolution in humans, because it's pretty much the same as it is in apes and monkeys, and in fact all mammals, in fact all vertebrates. So it was in a way invented by the precursor to all vertebrates. And then, as vertebrates speciated into more distinct families and more species, there was some tweaking and changes. But the immune system of mice is very, very similar to the immune system of people. So nature works not just by mutation and crossover, but a lot by copying. It found a good solution, it'll copy it again and again and again and start tweaking the copies. Okay. I should mention that you could have hypotheses represented by variable length strings. Okay? There's no reason you can't have that. In fact, nature does that with variable length genomes. When you do that, you have to worry a little bit about crossover. If you cross over two variable length hypotheses, the result would be of yet different lengths. So you need to make sure that you can interpret that. Again, it is all in your hands, how you define the hypothesis and how you define the rules to combine them. Just make sure that what comes out is interpretable by your system. So the slides have some examples of toy examples of how genetic algorithms solve them. I should mention one other important thing, and then maybe stop. You can do some math on genetic algorithms. You can try to prove some things, and the way that works is it's hard to say much about individual hypothesis, what happens in individual hypothesis, because the system is so stochastic, so randomized, that individual hypotheses are in the noise. But you can think of whole populations of related hypotheses. So they call them schemas. A schema is basically, you can think of it as a string, just like hypothesis, but it doesn't contain just ones and zero as hypotheses do. It also contains don't cares. So a string with these don't cares encodes the whole population of strings that fall into this template. So in one extreme case, you can have everything is don't care except one little area, and that would encode all the hypotheses that share that area, or in biological terms, share that gene. So you can now trace the stochastic behavior of this gene in the population. Now, let's define M as the number of instances in a population that fall into the schema. So that's the number of hypotheses in our currently held set of hypotheses, the current population, that fall into the schema S at time t. And now we can define a few other things. We can define F as fitness, and F bar is average fitness of the population at time t. We already defined M. Now we can define U hat as the average fitness of only the members of that schema. So think of the schema as, say, encoding a particular gene or a particular feature. U is the average fitness of those members of the population that have that feature or that gene, whereas F bar is the average fitness of the population. And it won't be surprising to you that if U is larger than F bar, chances are its representation population will grow. It will correspond to an advantageous gene or advantageous feature. So the probability of selecting an individual hypothesis is related to its relative fitness, which can be written this way. It's its fitness divided by the average fitness. But you can also talk about the probability of that the selected member would be not an individual hypothesis but come from that schema, and that can be represented as the ratio of the average fitness of the schema to the average fitness of the population times the current prevalence of the schema. And you can use that to write the expected prevalence of the schema in the next generation. It's expected because it's a stochastic process, and if the population size is small, there could be great variations. As the population size goes to infinity, this becomes a deterministic differential equation. So this is quite intuitive, but it neglects mutations and crossovers. Here's a complete schema theorem. This is the part we had before. It's the relative fitness of the schema times the current prevalence of the schema, but now it gets modulated by the other horrible things we do to it. This part is the crossover operator, so this is the fraction that's taken over by crossover, and this, if you look at it carefully, either in the book or in the slides, you will see where it comes from. It comes from the fact when you do a crossover, there is some probability that the crossover will cut in the middle of the schema. So that has to do with, is the schema small and is it concentrated, is it geographically in one area? If you cut in the middle of the schema, you're out of the schema, right? If the crossover cut in the middle of the schema, then you don't longer have that gene, or you have a smaller chance of having that gene. This part has to do with mutation, and again, this has to do with the probability that a random mutation will hit in the middle of your gene that you're interested in the schema, and would kill that gene or mutate it away. So you can go through the dynamics that you introduced in your rules of crossover, mutation, or any other rules, and calculate the expected value of different combinations of subpopulations, which we call schemas. I should also mention that the idea of genetic algorithms was used to search through a space of programs. And when you do that, computer programs, as hypothesis. And when you do that, the result is called genetic programming. So here's an example of a representation of programs that is amenable to genetic algorithms. So these are programs that calculate a numerical function. And you can represent a program as a tree with operators at the nodes, and any random tree would correspond to a particular mathematical function. And now you can imagine searching the space. Suppose you have a target function in mind that you're trying to approximate. You may not know what that function is, but given a candidate, you can evaluate the fitness of that candidate by maybe evaluating the mean squared error over some known training set. So now you can try many different combinations. I don't know, to me this doesn't make much sense intuitively as a way of going about doing things, but nonetheless you can do it in small toy examples. Well, you need to define crossover operations and mutation operations. So what would crossover be? Well, you can take two trees and choose an internal node in each one of the trees and swap the subtrees located in those internal nodes, right? That would be crossover. Mutation is simple. You can just take one of the nodes and replace the operator at that node with some other operator, into multiplication instead of division and so forth. And you may want to define other operators like splitting a node and creating another operation, whatever. Again, this is not how I would go about approximating a numerical function, but I guess it's a fun way of experimenting with genetic algorithms. Oh, here's a visualization of what a crossover would look like. So you see the two nodes that are chosen, and then they get flipped. Genetic algorithms were mostly demonstrated in toy domains, but there have been some non-toy areas, so I should mention them. I don't know all of them. There may have been quite a few since then. So here's an example of designing electronic filter circuits. So the individuals in the populations are programs that transform beginning circuit into final circuit. And you have a large population, heavy-duty computation. Genetic algorithms always take a lot of computation, so searching a large space. and you discover circuits that are competitive with best human designs. I'm always a little cautious when I see results that are competitive with the best available. I'm much more impressed if I see something that's better than what was done until then, because there are many ways of being competitive with the best. Many times you can bring into your formulation of the problem enough constraints that put you right there. So I think I gave you the example of classifying text documents by subject, and I showed you there were many ways of getting very comparable classification results. So if you can get better than the pack, that's worth noting. So as you see, I'm a little skeptical now. That isn't to say it's not a promising area. There are a lot of people who believe in it and push it. I'm not familiar with big success stories in machine learning involving genetic algorithms. It doesn't mean they don't exist. Questions? So, I'd like to devote the last few... Yeah, we do have a few minutes. Good. Last 10 minutes to ask you to fill the TA evaluation forms. Cut this?
Are you done?
Yeah.